Welcome back into this course. In the last session, we have discussed about the process of formation of the RNA from a double strand duplex DNA. This process was called transcription, which is a part of the central dogma of biology. Now, the next step, because once the RNA is synthesized, then the next question is what is the how the proteins are synthesized, and these proteins are synthesized by a process what is called translation process. Okay. So, translation is the process, or it is the final product of gene expression to make a polypeptide chain of amino acids whose sequence was prescribed by the genetic code. So, there are now certain things which we need to clarify. One is this important thing, the genetic code. Another is translation, which we already know that when we have the RNA, specifically now we have to say, I told you that what is the function of the three classes of RNA. The mRNA is actually translated, the information in the mRNA is translated into the protein. Okay. Now, what is the basically in protein we have amino acids linked together one after another. So, which amino acid will be linked to which the other amino acid, that will depend on the sequence of bases in the mRNA. Okay. And that is what is called the genetic code. Okay. So, genetic code is basically a 3-letter code. 3-letter code means that in RNA we have these 4 bases A, U, G, C. Now, the genetic code is a 3-letter code. That means, out of these 4 bases, you can by permutation and combination, what you can have, that you can have 64 combinations. You can have 64 combinations if you want to make a 3-letter code from these 4 letters. Okay, like suppose say one will be definitely A U G, another can be A G C. So, just keeping in the front, you can have A G C, then A, then you can change this A G U, that also can change, and then you can have A C G. So, like way the basically what is the number, ultimately how the 64 number is attained. Basically, you have 3 positions where you have to fill with these 4 letters, and there could be another important point, there could be repetition of the letters. That means, what is repetition? That means, you can have 3 A's also, you can have 3 G's, 3 U's, or you can have 2 A's, 1 C, all these things. So, to solve that, basically what you do, that for the first position you have, because there are 4 bases. So, you have 4 possibilities for the first, to fill up the first position. The second position also you have 4 possibilities, because you can have repetitions. Okay. And for the third position you can also have 4 possibilities. So, the total number of possibilities is 4 into 4 into 4, and that is called 4 cube, that becomes 64. Okay. So, these 3-letter codes actually code for a particular amino acid. That means, a single base cannot code for a particular amino acid, nor a double letter code cannot indicate or translate into an amino acid. It requires 3 letters, that means 3 base codes, letter means the bases in the RNA, 3 base codes that code for a particular amino acid. Okay. Now, we know that there are in proteins there are 20 amino acids. So, virtually what we need, the minimum number of genetic codes that we need is 20. That would have been sufficient, that one genetic code will translate for one amino acid. However, 20, the number you cannot reach, the 20 cannot be reached. If you take a 2-letter code, then your number of genetic codes will be 16. That does not encompass the whole 20 amino acids. So, nature had no other option to bring in a third letter, so that you have sufficient number of codes to cover up the entire amino acid spectrum. Okay. But while doing so, what now you see that you have many extra codes, right, many extra codes, because you have 64 basically. Now, these individual 3-letter codes are called the codon. Okay. So, genetic code is basically the whole alphabet structure, that which amino acid is expressed by which genetic which codon. Codon is the 3-letter, 3 base sequence that code for a particular amino acid. That is what is called a codon. Okay. Particular amino acid, that is what is called a codon. Now, how many codons are there? 64 codons. Okay. 64 codons can code for 20 amino acids, but the story is not complete there about the genetic code. Out of these 64 codons, there are 3 codons which are called stop codons or nonsense codons. Okay. Nonsense means that does not make any sense, that means that does not code for any amino acid. So, that is called these are called stop codons, and the rest 61 are coding for different, this is one or the other 20 amino acids. Okay. Now, let us just why this is important, because now we are trying to discuss the translation process. Translation process means how the mRNA, from the mRNA the proteins are synthesized. Okay. Once we have the, we say what is the once we have the protein, then the primary structure becomes very important, that which amino acid is connected to which amino acid. Okay. Different proteins have different amino acid sequence. Okay. And this amino acid sequence is dictated by the sequence of codons in the mRNA. Okay. I hope this is clear. So, mRNA you can divide it into several codons, and if it is transcribed into translated into the protein, then what you have, depending on the codon here, you have a particular amino acid, say 1, depending on the codon here you have the second amino acid 2, and depending upon the codon here you have the third amino acid. So, this way the amino acid sequence is varied by varying the different the codon sequences in the mRNA. Okay. Remember, the codon is present in the mRNA. So, the genetic code is the entire, all the 64 codons are like a periodic table, a table containing all the 64 codons, and then reading that you can tell that which amino acid is coded by which codon, because it is impossible to remember that if I say what codes for AUU, I think it is very difficult unless you are really going through the through that genetic table, that genetic code up till number of times, then possibly you may remember some things, but it is very difficult and it is not required also, because whenever we in periodic table it is still a little bit easier, or easier means for the chemists, because periodic table again if I say what is in the group 5. So, unless you are a chemist and handling the periodic table for a long time, you will not be able to tell what is there in the, you may tell the first one, but then the what is the second element, third element, fourth element, fifth element. So, rather than memorizing this, better check what is the genetic code. If you have that in front of you, you can tell okay, this is the code, this codes for this is the codon and this codon codes for this particular amino acid. Okay. Now, let us see what the table. This is the what I am talking about, the table, the genetic code. This is what is called the genetic code. Is now, in the genetic code, how this is read? See, you see all this UCAG on this side, then UCAG on the top side, and then you have several the other. See, each segment contains all these four nucleotide bases, okay, the 4 bases. Now, this is the now when I say AUU, suppose AAU, suppose you have to always specify which is the 5 prime end, which is the 3 prime end. Now, in this table, in most of the books it is written that this is the first letter, this is the second letter, this is the third letter. Letter means it is a base, okay, here. Now, the first letter is starting from 5 prime end and the third letter is ending at the 3 prime end. Okay. So, now how to read this table? So, if the first base is U, the second base suppose is U also, and the third base. So, this is the first letter. So, you have taken the first letter U, you have taken the second letter. Now, next you take the third letter. So, one of these you have to take. So, if you suppose I say what codes for U, remember this is 5 prime, this is 3 prime. So, that will be U. Then you see that what is written in the second in the second letter box, what is written under U, that means this one. This is UUU. This UUU is coding for phenylalanine. Okay. Suppose I say what is CCG, 5 prime to 3 prime. What codes for CCG? So, how do you go about it? So, you take C and then the next C, second letter is C, that means you have to take this one. So, C, C and G. Where is your C, C, G? That means you have to take here, this is your G. So, C, C, G. So, you will find here in the second column you will find CCG, this is CCG. So, CCG codes for proline. So, that codes for proline. Okay. So, this way you can remember one thing, that I can always try to fool you by giving some. Suppose I say what is GAU, but I put 5 prime here, 3 prime here. So, if that be the case, you always should do not be misled that you should not, this is your third letter, remember that, and this is the second letter and this is the first letter. Okay. So, from the 5 prime end the first letter starts. So, that is UAG. What is UAG? Let us see, this is U, this is A and G. So, UAG is here, then it is always, it will be always in the column of the second letter. Okay. UAG, that is a stop codon. Okay. So, that means this is a stop codon. So, that means as soon as you reach that UAG, that means the protein synthesis, whatever mechanism it is forming, that will stop at that point. Okay. So, now we come to the translation. Once we have that, first of all, where does translation takes place? Now, remember in a prokaryotes you do not have any compartmentalization. So, what happens here? Suppose this is your DNA. So, that is that will transcribe into the mRNA. So, suppose the mRNA is coming out like this, synthesized like this, it is coming out and into the cytosol, because everything is now cytosol, there is no compartment. So, the protein synthesis can take place before the whole mRNA comes out of the DNA machinery. Again I repeat that, maybe I can use another pen. See what happens, what I am saying, that if you have suppose the DNA, it is a big one and you are copying it into the mRNA. Okay. If you are copying it into the mRNA. So, suppose the mRNA is, suppose this is your template strand, this is your template, what happens, the red, suppose this is your change, that is not being changed. Let us see why the color is not changed. Yeah. So, this is the template strand and now the mRNA is synthesized from this end. Suppose this is your 3 prime and this is your 5 prime end. So, the mRNA is being synthesized from here. Suppose this is the initiation site. So, as the mRNA is synthesized, it is coming out of the system, okay. And then the synthesis is not complete yet, but because there is no separate compartmentalization of this process. So, wherever there is this protein making factory, which is called ribosome, by the way, ribosome is nothing but a protein making factory, which is also present in the cytosol. So, the ribosome will now start on this and will start making the proteins. That means, before the mRNA synthesis is complete, in prokaryotes, the ribosome can start working, and then synthesize the proteins, because this is the mRNA. So, from mRNA is only made because you need the protein from the mRNA. So, the protein is synthesized almost parallelly, okay. But in eukaryotes that does not happen, because in eukaryotes what happens, there are two things. One is, first of all, eukaryotes have a nucleus, a separate nucleus, where there is the DNA. Okay. So, when the RNA is transcribed, so it will more or less remain here, and once it is complete, then it is released in solution and then goes into the ribosome. This is suppose the ribosome, and the protein is now synthesized depending on the sequence of the codon sequence of the mRNA. Okay. So, that is one, that is the difference, but the reason why that cannot straight away go to the ribosome, because we will go into that, because in eukaryotes whatever mRNA is prepared, that needs to be processed so that we get the actual mRNA. I will come back to that point a few slides later. First, let us talk about the prokaryotic system. Okay. Now, what is the ribosome? How does it look like? Ribosome, actually basically there are two components of ribosome, two subunits. One is called a small subunit, which is the 30S subunit. Now, this is another, S means another unit of showing the molecular weight. This is a sedimentation rate. There is some unit which is the Swedberg unit, depending on the sedimentation rate of the system or the molecule that you are talking about. Okay. So, this is a 30S subunit and there is a 50S subunit. That means, there is a small subunit and there is a big subunit. Okay. And this is the large subunit, and the protein synthesis is taking place when both are actually held together, and that is what is called the complete ribosome, where the protein synthesis ultimately takes place. Okay. Remember, because this is not molecular weight, you cannot just directly add that the ensemble, that means these two things are held and the overall weight will be 50 plus 30. That does not happen here, because it is not proportional, it is not just addition of molecular weights, that 100 plus 200 is 300. It is not like that, that if you do a sedimentation of study of this, then you will see that that becomes 70 S. Okay. So, this is a 70 S ribosome in case of prokaryotes, little bit smaller than the eukaryotes. Eukaryotes ribosome is 40 S plus 60 S, okay. But it has also have a small and a large subunit, and the protein synthesis is done when the two are held together. Okay. So, this is the total ultimately becomes 80 S. So, the eukaryotes have 80 S ribosome, which consists of a 40 S small unit and a 60 S small unit, and in the eukaryotes you have 70 S ribosome, and that has got a 30 S and a 50 S subunit. Okay. You see that as this, what is the ribosome is made up of what? Ribosome is made up of many these are ribosomal RNAs. It is made up of RNA as well as protein, and there are many proteins in the ribosomes. If you take this eukaryote ribosome, the small subunit has 18 S RNA. Okay, this is called ribosomal RNA, and then 33 proteins. The large ones has 28 S RNA and 49 proteins. So, all these are held together and forms what is called the ribosome complex. Ribosome is nothing but it is a combination of proteins and ribosomal RNA. Okay. And this is one of the most important machinery in the cell, because you are making the proteins, which are basically the molecules which we require to sustain the living system. They actually work. Okay. The DNA has directed via RNA to synthesize the proteins, and the proteins are basically the ones which do all the job that are necessary. Okay. Mostly, there are a few exceptions. Okay. Now, what is tRNA? It says that tRNA, we know what is mRNA. mRNA contains the codons, okay, and that codons are specified. How are the sequences of the codons dictated? It is dictated, remember the mRNA is coming from the template strand of the DNA. So, if you know the template strand sequence, you can write what is the sequence of the messenger RNA, and you can now make out what will be the codon sequence in the mRNA, and from that you can write what is the protein sequence that will be made ultimately, the primary sequence of the protein. Okay. Now, that is mRNA contains the codon sequence, rRNA, the ribosomal RNA plays an important role and is present in the ribosome, that is the protein making factory, and then there is tRNA. The duty of the tRNA is to bring the amino acids one after another and then supply it, so that the protein chain grows one after another. Okay. How does the tRNA look like? tRNA has a structure like this. As I said that in these molecules there may be complementary inward, in the self complementary, like I showed you the hairpin. It is a single molecule, but that folds because one portion is complementary to the other portion. Similarly, the tRNA also has many complementarities, so that it ultimately takes a shape like this. It is a clover loaf type of shape. Now, this is the 5 prime region and this is the 3 prime region. And in the what happens in the 3 prime region, there is a wedge, definitely that will ultimately end with a wedge here, and this wedge is actually connected, finally it will be connected to like a smaller one, okay, through this A via acylation the amino acids are connected. So, the amino acids are basically forming an ester bond and it is attached to the tRNA, and these amino acids are actually being delivered to the growing polypeptide chain by the tRNA. Now, tRNA has another very important aspect, and that is at the this is the anticodon site, means when tRNA has to bind, we will go to that. tRNA has to bind to supply to the ribosome to supply this amino acid, but how does it know where to bind? That depends on the sequence here, and this is what is called the anticodon sequence. Anticodon sequence. So, the tRNA, now we know that tRNA has this acylated amino acid and it has got an anticodon. Anticodon means basically the opposite of the codon, but complementary, you can say it is the complementary of the genetic of the codon. Okay. And so, when there is this, yes, I think this is the what I was saying, that in the this is the shape of your tRNA, and there is this, why this takes a shape, because there is intramolecular hydrogen bonding like this. The 5 prime end is protected as a phosphate, 3 prime wedge is free, and that is where the amino acids are attached. This one like GAA, it is called the anticodon, and this whole loop kind of thing, that is called an anticodon loop. Okay. So, now let us go to the translation machinery. The amino acid attaches to the, as I already said, that tRNA can base pair with the codon of the mRNA at the anticodon on the tRNA. Let us see what does it mean. First step is, once the mRNA is synthesized, the tRNA also tRNAs are usually free, but they have to now hold up the amino acids via this acylation. So, charging is the process of covalently attaching the amino acid to the tRNA. So, each tRNA is covalently attached to amino acids. Now, which amino acid will be attached, that depends on the sequence of the anticodon. That means, if you have, this is your tRNA, the question is, if I say that which amino acid will be attached here, that depends upon what is the anticodon sequence here. Okay. Now, suppose this is your, I will write any sequence, say U A C. Suppose the anticodon is UAC, then what will be the actually the codon of this is the anticodon. So, the codon of this will be AUG. Now, if you go to the genetic code structure, the genetic code table, what is AUG? This is your A, what is your U, then U, and this is your G. Where is your G? Your G is here. Okay. So, AUG must be somewhere here, AUG. You see it is methionine. So, that means depending on this this one AUG, this is, I think, where is the other one, UAC. So, AUG codes for methionine. So, that gives an anticodon here. So, the tRNA, actually we had started with the reverse direction, that suppose the anticodon is UAC. Now, we write what is the codon for that, and we go to the genetic table and see that this codes for methionine. So, if this is methionine, then this will have a methionine attached to here. So, depending on the anticodon, the amino acid that will be taken up. Okay. So, the best way to do it, see the anticodon, and then if some question is asked that what is the amino acid that will be that will charge the tRNA, that depends on the anticodon sequence. So, what you do, you write the codon, according to you write the codon sequence, and then go to the genetic table and then genetic code table, and then find out which amino acid is attached to that. Okay. Finally, I think as eukaryotes, as I said, it is much more complicated. So, we will talk about the prokaryotes, and that will be sufficient for our course. Steps in translation. First, there will be initiation. What is initiation? The two subunits of the ribosome come together, and the start codon on the mRNA ribosome is aligned to set the reading frame. Okay. What is reading frame? Reading frame is that frame, that sequence of codons in the mRNA, that is called the reading frame. Then once these are all there, that means you have this small ribosome and the small subunit and the big subunit, and you have the your messenger RNA has been is attached to this ribosome, okay, bound to the ribosome. Then it has to start now. So, there must be a start. See where it will start, there must be a start codon, otherwise again the question comes that where does the protein synthesis start, which amino acid is added at the start. So, that is one question, and then stop codon. We already know that there are 3 codons which are stop codons. So, stop codon is not a problem. What is a start codon? Suppose this is a start codon, we will go back and then we will tell what is the start codon, but this is a particular codon which suppose this is the start codon. So, the amino acid synthesis, the protein synthesis now starts, okay. And the tRNA, will come here, one tRNA first comes here and binds here and bringing one amino acid here, okay. And next to it is another tRNA which comes here, and we will have another amino acid here at the 5 prime end. Now, there will be a reaction between these 2 amino acids. So, when the reaction takes place, this amino acid is transferred to that one, making this tRNA free of amino acid. Okay. And then what happens? The amino acid which now the tRNA which is free from amino acids, that will now leave, and this part will now come to occupy this part and leaving this other side free. So, another tRNA will come and then will sit here, and then the reaction takes place like this. So, I think I can explain it better by showing the diagram. See, basically here what is shown here. The ribosome, we told that there is a small subunit and a large subunit. Okay. So, initially the mRNA comes directly from your nucleus, in prokaryotes that is not the nucleus, it can directly come to the cytosol, and then what is not written here is that there is some binding, again there is kind of a promoter region, some binding sequence in the ribosomal RNA that is present in the small subunit. So, ribosomal RNA is present here, okay, and there is a proper binding region in the ribosomal RNA. So, the mRNA comes and now binds it. So, this is the binding point of the messenger RNA. By the way, this is your rRNA. Okay. So, that means here what is shown here, that first the mRNA, as I told you, mRNA binds to the small subunit, because there is a binding pocket in the ribosomal RNA present in the small subunit, and once that is done, then the large subunit comes and forms a complex. So, this is what the complex is called the ribosome, but now you have a combination of both small and the large. Now, the large subunit has I think this may I can make it little simpler and also much easy to understand. So, now you have I will write a shape like this. So, this is your small subunit. So, first the mRNA comes and there is a rRNA sequence which it recognizes, and once it recognizes, now it is the mRNA, right. This is your mRNA. As soon as that forms, now the bigger the other subunit of the ribosome comes, and now this is the actual ribosome where the mRNA is already bound. Okay. So, that means there is a sequence somewhere there where it is anchored. Now, the initiation, the mRNA has codons here, right. The mRNA has codons, and then according to the codons tRNA will come and bind, and tRNA remember has anticodon. Okay. This is little complicated, but you have to be very attentive here. Now, this ribosome has different sites. One site is called the P site, and another site next to it is called the A site. P site is nothing but peptide site, and this is the amino acid site, and on the left side of it there is something which is called an exit, E site, that is exit. Okay. Now, this suppose this is your first codon here. Okay. Codon is a 3-letter code. So, now according to this codon you will have a tRNA which will be bound to it. Okay. It will have a 3-RNA bound to it, and there will be an amino acid here, and the amino acid site there will be another, depending on the code here, another tRNA that will come, and bind to it. Okay. So, this is your OCO, one particular amino acid R1 NH2, and this is your OCO, another amino acid R1 and NH2. So, this is the situation. Now, let me tell you, this P site is always a consensus sequence, is AUG. What is AUG? AUG codes for methionine. That means, this is the binding point, and wherever there is now AUG, the synthesis will start from there. So, the methionine, this methionine containing tRNA will be the first one to bind to the P site, and then the amino acid site, the one of the amino acid will tRNA will bind depending on the codon next to AUG. Okay. Now, there will be a reaction between the two. So, this reaction is nothing but a transacylation. So, this NH2 comes here, attacks here, and this breaks, and this breaks. So, what does it mean? This means, so the ribosome, what I again, the small one, the large one. Okay. So, now you have the mRNA bound to it. So, first this is suppose the binding point, the location where it is anchored, then it sees where is your AUG from the starting point, from the where it is bound. Suppose there is AUG here. So, you will have a tRNA attached to it, I am not writing the full geometry, and there is another codon, but before that I told you that there are 3 sites. One is the P site, another is A site and another is E site on the left. This is exit, this is peptide, this is amino acid. So, there will be a amino acid here attached to it and there will be an amino acid here attached to it. Okay. These two are suppose different amino acids. So, the reaction that takes place is very simple, it is a transacylation. So, it was OCO R and then NH2 and next to it is another tRNA which will also have OCO, suppose R1 NH2. Okay, although they do not look proximity, but actually they are quite proximal and then this NH2 comes attacks here releasing the tRNA. Now, you see what happens, the product of this is that this is a free tRNA with OH here and this one has the protein attached to it. OCO, then you have first OCO, then you have the NH, this is CO, you do not O. So, you start OCO then R1, let us see again there is erase. Okay, just a second. So, you have OCO then R1, then what you have is NH, then you have CO and then you have R and then you have NH2. That means, you have now formed the first peptide bond. Okay. So, what happens now? So, you have basically now your this is the small, this is the large, this is your exit site, this is your P site, this is your A site. Okay. So, now after this at here is your free tRNA, because the methionine, that AUG which codes for methionine, that is now transferred into the A site. So, A site is now having this dipeptide OCO R, then NH, then CO, then this is methionine, I can write MET, it will be always methionine and then NH2. Okay. So, that is the scenario. Now, what will happen? Now, this is the mRNA. Okay. So, this is your starting point AUG, again I repeat AUG. Now, what happens? This ribosome will move one frame to the right side. So, if that happens, then as it moves, this will not, these tRNAs will not move. As this moves, that means, this will now come to the E site, that means, the free tRNA will come to the, this is the free tRNA, okay, RNA, because this is moving. So, this point is here now, P will be here. So, now P will hold the tRNA which is holding the dipeptide chain with the peptide, okay. In this case a dipeptide, and the amino acid site is free now, the amino acid site is free. So, now what will happen? Another tRNA will come, and which tRNA will come will depend on the sequence of the mRNA here. So, another tRNA comes and the same reaction happens, the NH2. So, you will get a tripeptide and then the ribosome slowly moves, like I think it may be somewhere here. See, AUG is the first one. So, this is the step where the tRNA, this is exit, that means, the free tRNA now goes out, okay. And leaving this, leaving the P site as the growing peptide chain. The first one is methionine. In this case, the example that is given here, the second one is proline. Now, this is your A site. So, this is the P site This is the A site. Earlier what was happened? It was shifted like this. So, this was occupying your P site at that time. Okay. So, as it shifts, the what happens? The growing peptide chain now goes to the P site, the amino acid site is free. So, now the next amino acid will be brought by the tRNA, and depending on the codon here. So, it is GUU, that means the anticodon will be CAA, CC, here it is GGU. So, that will be CCA. So, now CCA you have to see what codes for GGU, definitely what is written here glycine. Okay. So, that will come here and the next reaction will take place. The NH2 of this glycine will attack the peptide bond here attached to the proline, and the whole peptide will transfer to the right side. Okay. And then this also starts again moved one frame, means one codon, it will shift to the right and then crossing one codon, and then what happens all the time, there will be an amino acid which will be, there will be a tRNA which will be free. Now, that will come to the exit site and that will go away. So, in this way the protein synthesis takes place. So, this will continue up to which point? Up to the point till a stop codon comes. So, once a stop codon comes here, no tRNA will bind, and then the system knows that that is the end of it. The whole protein is released. Okay. Now, this I think it is also shown here, the growing polypeptide chain. This is the P site, this is the A site. You see the A site that is coming. So, the fourth one is added to the third one. So, while doing that, it is shifted to the right side, then it moves. Okay. I think that is once you go through this slide, it will be even more clear. I have explained it. Now, there is some questions, what is the, that means whether all proteins do not have methionine as the N terminus, because this translation process demands that all proteins, because the starting point is AUG which codes for methionine. So, the question is whether all proteins have methionine as the N terminus, that is not it, because many proteins have different N terminus amino acid. So, what happens that translation tells you that the methionine should be at the N terminus, but there is another process which is beyond translation, that is called post translational modification, that if methionine is not required as the N terminal amino acid, then there are enzymes which will take care of this process, the methionine will be removed and the correct protein will be made. Okay. So, that is called post translational modification. Now, that is but for your eukaryotes there is some problem. The problem is that in prokaryotes whatever messenger RNA is coming, that is completely converted into the protein. There is no other processing needed in between, but in eukaryotes what happens, the mRNA that is transcribed from the template strand of the DNA, that is what is called pre-mRNA. Why pre-mRNA? Because this mRNA is made up of, definitely all these codons are there. However, all the portions of it does not code for the protein. In case of prokaryotes, the full mRNA, suppose this is your start codon and this is your stop codon. So, the protein will be made according to this, the codon sequence, but in case of eukaryotes, the pre-mRNA will have to be further processed to make what is called a mature mRNA. What is mature mRNA? Where the full portion from start to stop is transcribed, is translated into the protein, stop and then start and then stop. Then this portion will be translated into the protein. This is not. because this contains some portions which are not passing any information, which does not code for any amino acid. Okay. So, we will in the next lecture we will do that, we will describe how it is done. Thank you.